My name is Igor. I'm a physician scientist. I co-direct with the Greenberg Pancreatic Cancer Institute. My role as a physician scientist is to see the patients and develop new ideas for treatment of pancreatic cancer. Right, so what is concerning to me about pancreatic cancer is that its incidence is rapidly rising. The reason for that rapid rise is not clear, but it is alarmingly becoming the third leading cause of death. We see increased incidence of pancreatic cancer in younger individuals. The fact that we see more and more patients with this cancer raises the urgency to develop new treatment options, which are currently very limited. This is probably one of the most refractory and aggressive type of human cancer that we know. So with the limited number of options that are available for patients with pancreatic cancer, we really focus at this point on understanding the cancer biology and what the vulnerabilities in this disease. And that creates the opportunity to apply certain treatments and certain medications based on the cancer mechanism. It is unfortunately the majority of pancreatic cancers do not carry druggable or drug amenable, I should say, mutations. However, there is a subset of tumors that do. And even for these patients, the treatment options are not indefinite. So any opportunity to develop new drug, new treatment modality, new mechanism that we can exploit for therapeutic gains to improve their quality of life, cure rates, longevity will be highly beneficial. One of the potential vulnerabilities in pancreatic cancer is deficiencies, either somatic or inherited in DNA repair pathway genes. This has been a focus of extensive research recently, with new drugs coming to the market and to clinical use. And this is one of the opportunities that we're exploring in the laboratory and also in the clinic. The search for genetic vulnerabilities in pancreatic cancer continues, and one of the opportunities that came about to us was this collaboration that we established with Lantern Pharma. Lantern has been a long time focused on exploring the opportunity to develop previously abandoned drugs because of the lack of understanding of their molecular mechanisms. And they brought to our attention this compound called LP-184, which is a precursor prodrug that will be converting to an active alkylating DNA damaging agent upon exposure inside the cells, inside the cancer cells, by an enzyme called prostaglandin reductase. So that immediately was of interest to us, and we began a very productive collaboration, which led to not only to publications but also to launching a phase 1 clinical trial, which we're very excited about. Let me tell you in a very few words what LP-184 is and how it works. It is a prodrug. It's a derivative of acylfulvene, an old drug that has been in clinical testing in 1990s. Lantern developed a more advanced version of this drug with improved pharmacokinetics, and we found through genetic manipulation that it absolutely requires this prostaglandin reductase, or also known as PDGR1, to be converted to a fully activated, rapidly acting DNA damaging agent. In the absence of PDGR1, these cells are completely insensitive to the drug, whereas high expression of PTGR1 confers increased susceptibility. And that offers yet another point of vulnerabilities, because it turns out that PDGR1 is expressed at high level in many tumors, including pancreatic cancer. We also found one of the potential mechanisms how PTGR1 can be induced. It happens through oxygen deprivation mechanism called hypoxia. When it's upregulated, it becomes a point of vulnerability to the cancer cells. So not only damages DNA, so tumors that carry mutations that confer deficiency in DNA repair pathways, and also high expressing a PTGR1 at high level are projected to be particularly sensitive to this drug. So we're very excited about the opportunity to find which patients are likely to benefit, whether the drug works, and whether we can provide an additional treatment option for patients with this devastating malignancy. So this is a phase 1 trial. It is designed to determine what is safe, whether patients can tolerate this chemotherapy drug. And we also are looking very attentively into a possibility that we find a subset of patients who might be potentially responding to the treatment, because we know the mechanism, we know the biomarkers of that would predict the response to this drug. We will look across multiple solid cancer types, including pancreatic cancer, where both PTGR1 and the incidence of DNA repair pathway mutations are frequent. So these are the areas of interest for us in both clinical development and also to understand the translational mechanism and the biomarkers. The patients we're looking for are the ones that whose tumors carry mutations in DNA repair pathway, because the mechanism of the drug is DNA damage. We also are looking for patients who would express high level of PTGR1. From our preliminary bioinformatics exploration, there is a large variety of solid cancers that express PTGR1 on high level, including pancreatic cancer and other solid tumors. So this is a phase 1 trial that will be open to any patients with incurable solid cancer. One other potential consideration is for patients with glioblastoma, because of the history of use of alkylating agents. In fact, temozolomide is the only FDA-approved alkylating agent that is currently widely used to treat this disease. We also know that glioblastoma expresses high level of PTGR1 that converts LP-184 to inactive compound. And I think for many of these two main reasons, should be some level of enthusiasm in GBM to see whether this drug has any activity. It has a good pharmacokinetics and ability to penetrate to the brain. From my experience in serving as a medical oncologist, I know that oftentimes people and my patients live as long as they have options for treatment. So we are actively searching for clinical trials. I'm trying to do my best to help my patients to find suitable clinical trials for which they can be a good candidate. And it's, this kind of exercise is reliant on understanding the cancer mechanism. So I do firmly believe that we will advance and improve the cure rates, and our options for treatment will increase once we put more patients on clinical trials. So anyone who is a patient or who has a family member affected by pancreatic cancer or any other devastating malignancy should be interested in understanding clinical trials, why we do them, and whether he or she can be involved in clinical research and enroll the patient to this clinical experiment. These are needs that are urgent and absolutely critically important. And there's always, I firmly believe that there's always room for a miracle. If we match the drug with the mechanism of the tumor, we can win.